Hi, and welcome to Optimum TV. My name's Stephen Adams, and today I'm joined by Stefano Gulla, CSO of Amsterdam-based Kling Therapeutics. Hi, Stefano.
Hi, how are you, Stephen?
I'm good, thanks. How are you?
Good.
Good stuff. Kling has developed a platform for discovering antibody-based drugs based on interrogating the immune system of exceptional responders to disease. But Stefano, you're the expert. Tell us a little bit more about your technology.
Hi, Stephen. Our technology, the core of our technology, is our ability to immortalize primary B cells from patients. You mentioned elite responders as our source. So we look for patients who have had an exceptional immune response to infectious disease or even in the context of oncology. And by applying our technology, we can generate these living libraries of immune cells, B cells, that we can then use to interrogate and understand what the immune system is recognizing that can allow us to produce new medicines based on that.
And your platform can interrogate lots and lots of these B cells from elite responders at once. Is that right?
That's correct. This is one of the power of the technology itself, is the efficiency by which we can immortalize these B cells. It allows us to capture over 90% of the immune repertoire of a particular individual. And that itself allows us to then be able to ask very detailed questions about what is the immune system actually recognizing. This is how we come up with our very unique and novel targets, either for tumors or infectious diseases, which are the underlying basis of our platform, as well as our pipeline as we're developing it.
And the idea of using B cells from elite responders, I'm presuming that the idea behind that is that if you've got to look for a needle in a haystack, you might as well look in the right haystack.
Well, that's correct. We know that there are individuals whose immune system is able to recognize unique targets, either by the fact that they can identify epitopes on those targets that are novel, or they identify unique antigens that are associated with a response. For example, in the case of infectious diseases, we know that there are people who are naturally able to develop antibodies that can fight an infection much more effectively. And these are the people that we want to go after, understand their immune response, and potentially then use these antibodies as the basis of new therapies.
You mentioned infectious diseases. I know that Kling is also looking for targets in cancer. Are those the two main broad indications that Kling is looking at, or are there other potential disease areas too?
The technology itself is disease agnostic. In that context, though, we are interested in diseases where the immune system has a fundamental role. So clearly, in the case of infectious disease, that's an obvious choice. We know the immune system primary function is to fight infections. And more recently, we, as a field, we have understood more about the role of the immune system in the context of oncology and response to cancer. So this is also another area where we're interested in using our platform. Moving on beyond that, there's now emerging evidence of the role of the immune system in neurodegenerative diseases. And that's an area where we'd like to expand in the future as well.
Okay, interesting. Tell me, has the fundamental technology on which your platform is based had any notable hits in terms of drug development in the past?
This is very unique for a company of our stage. Our technology, in fact, has been applied in several different cases and been able to derive many hits. In the case of RSV, we have actually discovered an antibody that's now been marketed, which is the basis for the drug Beyfortus. This is an antibody that's being used to treat children that that are potentially being exposed to the RSV disease, and has been shown to be very effective in actually doing that. So we're very proud of the fact that even at this early stage, we've been able to bring forward a life-changing medicine for these patients.
Okay, and tell me about your current lead program or lead programs.
We have a clinical program, KBA 1412, which is an antibody targeted to CD9. This antibody was derived from a patient who has had a curative response to a melanoma, advanced stage melanoma. What's unique about this antibody is that it's able to recognize a particular epitope on CD9, which we find is able to circumvent some of the previous challenges that had been described for targeting CD9. We're now advancing a phase 1 clinical trial in solid tumors, and we're very excited about the results that are coming out of that.
Excellent. Well, that leads me on to my final question. What does Kling hope to achieve in the next year or two?
We want to, and we are on our way to establish Kling as a premier antigen discovery company. We believe that we can use our technology to derive many more insights into which antigens are more important to target for both infectious disease, for cancer, but also, as I said, potentially neurodegenerative disorders. And as we've seen over the past years, there's been an explosion in the amount of technology that has gone into producing even more effective drugs with protein engineering, such as bispecific antibodies, ADCs, CAR-Ts. We believe that our technology is able to further complement this technology explosion by adding novel antigens that can be used and leveraged with the technology that are being developed now.
So that suggests you might be able to work in partnership with some of these companies using these types of technologies?
Absolutely. I think partnership are the basis of the biotechnology industry, both with other biotechs as well as with pharma. There's a lot of technology out there that can be used in conjunction with our discoveries in order to advance novel therapies. But we also are looking forward to advance our own pipeline, both in infectious disease and oncology. So we're open to a number of potential discussions at this point.
Stefano Gulla, thank you very much indeed. It's been great talking to you. Thanks everybody for watching. Bye-bye.
Thank you very much, Stephen. Bye.